Hello everyone, I welcome you all to Biology Lessons with Kalyani. In today's class, we will be discussing about a new chapter, that is cell cycle and cell division. This is the part 1 of this lecture series in which we will talk about mitosis, and part 2 I will be uploading shortly, wherein I will discuss about meiosis and its different stages. Now let's look into what are the different things we will discuss today. So first we will look into why cells divide. Then we will discuss about what is cell cycle and what are the different stages of a cell cycle. Then we will talk about cell division, its types, and we will cover mitosis in detail. We will talk about its different stages, and then finally we will discuss about the biological significance of mitosis. If we consider a cell, only way in which we can make a copy of it, okay? Now consider, let's say we have a cell, okay? Only way in which we can make a copy of it is by duplicating the pre-existing cell, okay? So this cell has to duplicate itself to make copy of itself, right? So it is the only way in which new cells can be formed, okay? And the genetic information, whichever is stored in the nucleus, can be passed on to the progeny, okay? Now by duplication, I mean there will be duplication of the genetic material, right? As well as whatever organelles are there and the macromolecules are there, that has to be also multiplied or duplicated so that it can be passed on to the progeny in equal proportion. Is that clear? So cell division means duplication of the pre-existing cell, and it means DNA replication plus cell growth, and ultimately leading to the synthesis of new daughter cells, right? Now an overview of the different types of cell division. So we have two main types of cell division: mitosis or equational division, in which if we compare the parent cell and the daughter cell, both will be genetically identical and chromosome number remains same. And next is your meiosis. What is meiosis? Meiosis is the reductional division. Suppose we have a parent cell which has a 2n chromosome number. In meiosis, we will have the daughter cells which will be having half of the chromosomal number. So mitosis is equational division, meiosis is the reductional division, and mitosis and meiosis are the two main types of the cell division, right? Now what are the functions of cell division? So this cell division is important for cellular repair, and wherever there is old tissue or injured tissue, we want to replace it. So their cell division is very important for cell growth, okay? Then you have something called as formation of the gametes. Now in sexual reproduction, we know that gametes are formed which fuse together to form a zygote. So for gamete formation also we need cell division. Now this cell division can also be a mode of reproduction. In which kind of organisms? In case of your unicellular organisms. So basically this cell repair, replacement, cell growth and reproduction, they all take place by your mitotic cell division, and the gamete formation, it undergoes meiosis, right? We will know more about it in the upcoming slides. It has been seen that the cells, they divide only when there is a disturbance in their nucleocytoplasmic ratio. What does that mean? Nucleocytoplasmic ratio means volume of nucleus divided by volume of the cytoplasm. Let's say this is a cell. This is its nucleus and this is its cytoplasmic content. Now, as a cell takes up the nutrients from the surrounding environment, it increases in size. By increase in size, I mean that there is a growth in the cytoplasmic content of the cell. So what happens? This ratio is disturbed. Why? Because the denominator, that is the volume of cytoplasm, is more. So when the nucleocytoplasmic ratio is disturbed, the cell tends to divide. Is that clear? Now let's see what is cell cycle. Now I already told that when a cell divides, there are two things which happen. First is cell growth, in which there is the increase in the number of the organelles and the macromolecules, whatever we need, that is synthesized. Why? Because that has to be passed on to the progeny. Second thing is that the DNA content is also doubled, because we need to divide need to divide everything equally so that it can be passed on to the progeny. So consider this is your cell. There is first a cell growth stage and the chromosome duplication stage, that is called as the interphase. Okay, so whatever is there in the cell, the content of it is essentially doubled here. Okay, next thing is that the actual multiplication stage. Okay, this multiplication stage, what is happening, the cell is actually dividing. So it takes place in two phases. First, the nucleus is divided, that is called as the karyokinesis. Then the cytoplasm is divided, that is called as the cytokinesis, leading to the formation of two daughter cells. Is that clear? So the cell cycle is a series of events as the cell grows and divides. So as the cell grows and divides, whatever events are taking place, that is all comprised in the cell cycle. Now it consists of two stages: preparatory stage plus the actual division stage. Now this preparatory stage, in which the cell growth and the chromosome duplication is happening, it is called as the resting stage or the interphase. By resting stage, I don't mean that the cell is metabolically inactive. It just means that the cell is not dividing. It's preparing itself for the division in subsequent stages. Right? Now, multiplication stage has two parts in it: karyokinesis, which is a division of the nucleus, and cytokinesis, which is a division of the cytoplasm. Clear? This cell cycle is very much coordinated, and it is genetically controlled by different proteins such as cyclins and cyclin dependent kinases. Also, it is very sequential and ordered in nature. Typically, it has four stages, that is interphase, which comprise of G1, S and G2, followed by the multiplication stage. Now let us look into the different stages of cell cycle in detail. So the cell cycle is the entire series of events that a cell undergoes, during which it divides, okay, and forms the new daughter cells. It essentially consists of two stages. What are they? Interphase plus actual dividing stage. Now if you see, the portion marked in blue is the interphase. It is the longest stage of the cell cycle, and it constitutes 95% of time of a cell cycle. This interphase is also called as the preparatory stage as well as a resting stage. Why it is called resting? That the cell does not divide, but it doesn't mean that it is metabolically inactive. The cell is very much metabolically active in the interphase stage. It's just that the cell does not divide. Okay. Now, interphase is divided further into three stages: G1 phase, S phase and G2 phase. Now, G1 phase is also called as the gap 1 phase. In this stage, typically the many different proteins are synthesized. There is an increase in cell size as well as the cytoplasm. Most of the organelles as duplicated. Now, exceptions of this are chloroplast, mitochondria and Golgi body. Apart from that, other organelles are duplicated in the G1 phase. Now, if you see, this G1 phase is the longest phase of the interphase as well as the cell cycle. Okay, very important question. Longest stage of the cell cycle is G1 phase. Okay, in whole it is interphase, and if you compare all the stages, among them G1 is the longest phase. Now, if you see the location of this G1 phase, it is placed between the M phase, that is a dividing stage, and it comes before your synthetic phase. That's why it is called as the pre-synthetic stage. Also, it is called as the post-mitotic phase. Now, question can be asked like this: the interval between the cell division, okay, and the DNA synthesis is called as the gap 1 or the G1 phase. So, you should pay attention to the order here. So, that's all about the G1 phase. Let's look into what is the S phase. Now, S phase is called as the synthetic phase. Because it comes after the gap 1 phase, it is called as a post G1 phase, and as it comes before the gap 2 phase, it is called as a pre G2 phase. So in this S phase, typically what happens is that the content of the DNA, it duplicates. So it is the actual stage in which DNA is replicated, or two copies of the chromatids are formed. Or if you see, first the chromosome exists as a single thread, so it synthesizes the sister chromatid in your S or the synthetic phase. Also, very important in this stage, centriole duplicates, and one very important protein, that is the histone protein, is formed. Okay, now typically this S phase takes place both in nucleus as well as cytoplasm. So in nucleus, what will happen? The DNA content is getting increased, and in cytoplasm, centriole duplication takes place and histone formation takes place. So often question is asked like, centriole duplication takes place in synthetic phase. Okay, now the DNA content is only increased in the S phase, but the chromosome number does not change. That's a very, very important point which you need to remember. The chromosome number remains same, only sister chromatids are formed. So if you say the initial DNA content is 2C, it becomes 4C after the synthetic phase. Let's see the third phase or the final phase of the interphase, that is called as the G2 or the gap 2 phase. Now this G2 phase, because it comes after the S phase, it is called as a post-synthetic phase, and as it comes before the division phase, it is called as a pre-mitotic stage. Now this is the interval between the DNA synthesis and the cell division. So pay attention to the order. If first DNA synthesis is asked, then the cell division stage or the multiplication is asked, that is called as a gap 2. But if otherwise, like if it is changed, is the interval between M or the multiplication stage and the S phase, if it is asked this way, then the answer will be the gap 1 phase. Okay, so let's come back to the G2 phase. This G2 phase is very important because here all the other organelles which were not synthesized in G1, they are synthesized here, such as mitochondria, chloroplast and Golgi. They duplicate here. One very important protein, tubulin, is synthesized here. Why? Because you need microtubulin for the formation of the spindle fibers. So it is just ready before your actual cell division starts. Okay, now this is your mitotic phase or the M phase or the multiplication phase. This typically consists of 4 steps of karyokinesis or the nuclear division, that is prophase, metaphase, anaphase and telophase, followed by a cytokinesis stage, which is the division of the cytoplasm. Okay, I hope this cell cycle is very clear. Just another important thing which you need to remember is this cell cycle duration, it varies from different species. So if you take humans, it is 24 hours. If you take yeast, it is 19 minutes. Okay. Now, this time which the cell needs to make new copy of itself, or time it takes to divide, it is also called as the generation time. So generation time and the cell cycle interval, it is also sometimes used interchangeably. I hope cell cycle stages are clear for you now. Next, let's look at the cell checkpoints. So we have this cell cycle, which is a very important stage, and we need to check whether all the steps are taking place very properly or specifically or not. If there is any error in the different stages, there will be an error in the chromosome number, so we cannot afford that. So cell has different checkpoints at important junction between the phases, so that the proteins that are responsible for it are typically cyclins and the cyclin dependent kinases or the CDKs. So most important checkpoint is the G1 to S checkpoint. So what happens in this checkpoint is that the proteins check whether the cell needs to enter the cell cycle and proceed to S phase or not. Okay, so if if a cell, it has to undergo cell division, it has to pass the G1S checkpoint. Okay, so in this stage it is checked if the environment is favorable for the cell to divide. If the cell passes this particular checkpoint, then the cell is destined to divide. Okay, so in this there is an important point. So there are some cells which do not pass this G1S checkpoint. They tend to remain quiescent, or they tend to not divide. So they enter something which is called as the G0 phase, or it is also called as the quiescent stage. Okay, this is also known as the quiescent stage. So what happens? The cell exits the G1 stage here. The cell exits the G1 stage. The cell does not exist the cell cycle. It just exits the G1 stage and becomes temporarily inactive. What do I mean by inactive? The cell is metabolically active, but it just does not divide. Okay. Example of it can be heart cells. And if you consider plant meristematic cell, so they are also example of it. So if the meristematic cell in case of plants, they tend to divide always. Okay. Now, if you consider the meristematic cells in case of plants, they tend to divide always. Okay. Sometimes what happen, certain cells, they decide not to undergo cell division any further. So they become permanent cell or parenchyma. But under certain situations, whenever the cell needs, or there is a cell repair which is needed, that particular cell can be called back from its D0 phase again to dividing stage. So here in case of plant, it becomes the secondary meristem. Okay, it becomes the secondary meristem. So about G0 phase or the quiescent stage, we should remember that the cell becomes temporarily inactive or does not divide, but it can be called back again to cell cycle and can be lead to further cell division. The cell exits which stage of the cell cycle? Is exits the G1 stage of the cell cycle. That is, it does not pass the G1S checkpoint, but it can be always called back to the cell cycle. Example can be heart cell and the meristematic cell, which form the secondary meristem. Okay, let's see another checkpoint here, that is the G2M checkpoint. It checks whether everything goes on fine with the your mitosis stage. And there is another important checkpoint, which is the metaphase anaphase checkpoints, which checks whether the chromosomes are divided equally or not. So these three are the different checkpoints. Among them, G1S checkpoint is the first very important checkpoint which decides whether the cell should divide further or not. So let's now go to the actual division stage, or the mitosis, or the M phase. Now this mitosis stage, it takes place in case of the diploid somatic cells. Okay. Now it has been seen that certain haploid cells also undergo your mitosis. Generally, mitosis takes place in the diploid somatic cell, but exception are there. Exception of this is the male honeybee or the drones, in which the mitosis takes place in the haploid cell. Now, in case of plants, if you see, mitosis takes place in both haploid and diploid cells. Now, in haploid cells, mitosis can be seen in algae and bryophytes, while in diploid cells, mitosis can be seen in the higher plants. Okay, mitosis is also called as the equational division. Why? Because the number of chromosome does not change in case of mitosis. Now, what is the significance of it? The significance of mitosis is growth. Wherever we need growth of new cells, and we need to repair the cell. For example, cells of the upper layer of epidermis, cells of the lining of the gut, or the blood cells that are constantly replaced with the help of your mitosis. Now, this mitosis is also helpful to restore the nucleocytoplasmic ratio, and it is very important wherever we need to make genetically identical cells. So these are all the significance of mitosis. Let us now discuss the mitotic phase in detail. Now, we already have seen that the cell which was in interphase, it has synthesized most of the proteins, cell organelles that it needs for division. And in the S phase, we know that the DNA which was present in the form of single chromatid, it has doubled and a sister chromatid has been formed. But in the interphase, if we see, the chromosomes are present as very thin coiled like structure. Now consider a cell, and we are trying to divide its content. So if the chromatin, which is your DNA or the main genetic material, if that is tangled up, we cannot easily divide them equally. So what do we need first? So first we need the condensation or the untangling of that DNA. So in the first stage of the mitosis, what happens, that is your prophase stage, there is condensation of the chromosomal material or untangling of the chromosomal material. Okay, so see, this is your initial prophase, and by the end of the prophase, the chromosome, it has considerably thickened up and we can individually see them. Okay. Now, next what happens in prophase is we need something which will help us in division of the chromosome. So here what happens, the centrioles, which was also duplicated in your S phase, that plays a role. The centrioles, we know that they form the spindle fibers, right? So this pair of centrioles, they move to the opposite pole in the prophase and they radiate what is called as the spindle fiber. Okay. So these spindle fibers will further attach to the chromosome and help in the division of the chromosomes equally in subsequent cell stages. So let's see what are the characteristics of prophase. We already discussed initiation of condensation, untangling of the chromosomal material, movement of the centrosome towards the opposite poles. The chromosomal material condenses to form compact mitotic chromosomes. You can see here in late prophase. Chromosomes can be seen now composed of two chromatids attached together at the centromere. Each centrosome radiates out microtubules called as the aster. So this centrosome, it will radiate the microtubules and those microtubules can attach to the chromosomes. Okay, this centrosome is called as the aster. The aster together with the spindle fiber is called as the mitotic apparatus. So what is mitotic apparatus? Two centrosome along with the two centrosomes that are called as the asters along with the spindle fiber is together called as the mitotic apparatus. By the end of the prophase, if we see, we do not need the nuclear envelope. Why? Because we need the chromosomes to spread out nicely in the cytoplasm. So the nuclear envelope is disintegrated. Also, organelles such as Golgi body, endoplasmic reticulum, nucleolus, they also disintegrate. Okay, so these are the characteristic of the prophase stage. Metaphase is the second stage of mitosis. It is characterized by chromosomes that lie at the equator. Okay, all the chromosomes are highly condensed in metaphase. The condensation of chromosomes which has initiated in the prophase stage is completed in the metaphase. Okay, and nuclear envelope disintegration, which has also begun in prophase, that is also completed in the metaphase. All the chromosomes, they lie in the cytoplasm and they can be seen present at the equator in a line. Okay, that imaginary line is called as the metaphasic plate or the equatorial plate. Each of the chromosome can be seen attached to the spindle fiber radiating from each aster. So one chromosome is attached to two spindle fibers. Okay, the asters or the centrioles which are present in the opposite pole help by moving the chromosomes towards the poles. So each chromosome, the centromere of it is attached to two spindle fiber, one from each pole. Okay, so if you consider humans which has 46 chromosomes, there will be how many spindle fibers present? 46 into 2. Okay, so each chromosome is attached to two spindle fibers, right? Now, where is this spindle fiber attaching itself to the chromosome? If you see the structure of the condensed chromosome in metaphase, it has something called as the centromere, its chromatids. Okay. Now, in the centromere, it has a protein disc which is called as the kinetochore. These kinetochores serve as the sites of attachment of the spindle fibers. Right, so the chromosomes that lie at the equator and the plane of the alignment of the chromosome is called as the metaphasic plate. This metaphase is widely used to study the morphology of the chromosomes, right? So that's all about the metaphase. Let's see the third phase of the mitosis, that is the anaphase. So now all the chromosomes were lying at the equator. So what happens? The spindle fiber is attaching itself from both the asters to the centromere. These spindle fibers will try to pull the centromere. So as a result of it, what will happen? The centromere split. Okay, one centromere is being attached to two spindle fibers from opposite poles. So the spindle fiber will contract and try to pull the centromere towards each other. So see, this is the centromere and this is the kinetochore, and the spindle fibers, they are attached to two ends and they are trying to move in opposite direction. So as a result of which, what will happen? The splitting of the centromere takes place. So one part of the centromere will have one sister chromatid and the other will have the other set of the sister chromatid. So in anaphase, centromeres split and chromatids separate. The chromatids, where do they move? They move to the opposite poles with the help of the spindle fiber. The centromere of each chromosome remains directed towards the pole. The centromere will be directed towards the pole, and the chromatids will be trailing back. See, centromere towards the pole and the chromatids trailing back. This is the best phase to study the shape of the chromosome. Okay. So this anaphase is characterized by each chromosome arranged at the metaphasic plate is split simultaneously, and the two daughter chromatids, now referred to be as the daughter chromosomes of the future daughter nuclei, begin their migration towards the opposite pole. So anaphase essentially is characterized by movement of the sister chromatids towards, or the daughter chromatids towards the opposite poles. Clear? And splitting of the centromere. Is that clear? I hope anaphase is clear now. Let's look to the fourth stage of the mitosis, that is the telophase. Now, telophase essentially is the reverse of prophase. Chromosomes that have reached their respective poles now decondense. Now, in telophase, you cannot see the thick chromosome typical structure. They all decondense, they tangle up and lose their individuality. The individual chromosomes can no longer be seen, and each set of chromatin material tends to collect each of the two poles. Okay, this becomes a nuclear region and the further future nucleus of the one of the daughter cell, and this is the future nucleus of the second daughter cell. Nuclear envelope also develops around the chromosome and clusters at each pole, opposite of the prophase, right? Nuclear envelope develops, nucleolus, Golgi complex, ER also reform. These organelles have, disintegrated in prophase. They come back in your telophase stage. Okay. So division of the nucleus is complete. Next, we have to divide the cytoplasm. So what happens? Cell itself is divided into two daughter cells by the separation of the cytoplasm, and that is called as a cytokinesis. So first, what happens? Division of the nucleus or the karyokinesis takes place. After the nucleus is divided, then there is division of the cytoplasm. Okay. That process is called as cytokinesis. So that what happens in case of animal cells, the cytokinesis is achieved by appearance of a furrow. See, there is a furrow which moves from outwards towards the inside. The furrow gradually deepens, ultimately joining in the center to lead to the formation of two daughter cells. But in case of plant cells, what happens? There is presence of cell wall, which is a rigid structure. This furrow cannot take place. So what happens? In the middle, there is something called as a cell plate. In plants, wall formation starts in the center of the cell. Center of the cell, there is a cell plate formation. The formation of the new cell wall begins with the formation of the simple precursor called as a cell plate. So this moves from inside to outside, forming the new cell. Sometimes what happens in certain cells, the nuclear division takes place, but there is no division of the cytoplasm. So if the cytoplasm doesn't divide, what do we get? We get a multinucleate cell condition. So that type of cell, cells in which karyokinesis takes place but no cytokinesis occurs, we get a formation of something called as syncytium. An example of it is your liquid endosperm of coconut. Very, very important. Okay. So that's all about cytokinesis. So let's just quickly revise the different stages of mitosis. So this is your cell which is in interphase. In interphase, what happens? Cell growth, protein synthesis, synthesis of the cell organelles, okay, duplication of DNA in S phase, okay. Interphase is also called as your what? Resting phase or the preparatory phase. This is your dividing stage or the mitosis. So first stage is what? Prophase. What happens in prophase? in the prophase, the nuclear material starts to condense, and then nuclear envelope also starts to disintegrate, and organelles such as your Golgi body, endoplasmic reticulum, nucleolus, they start to disintegrate. Then the pro-metaphase, there is condensation of the nuclear material which completes in the metaphase, and the chromosomes lie along the equator. They are attached to the spindle fiber through the kinetochore, and each chromosome is attached to two spindle fiber. Okay, now this metaphase is best to study the chromosome structure. Then this is your anaphase, in which the sister chromatids, they are pulled apart to the poles after the splitting of the centromere, right? Now in the telophase, there is formation of the, the nuclear material decondenses, and the chromosomes lose their identity. The nuclear material pulls towards the opposite pole to form the future nucleus of the daughter cells. Then this, after the nuclear division, there is division of the cytoplasm or the cytokinesis. So that's all about the mitosis stages. Is that clear? Now we have already discussed about the different stages of mitosis. Let's just discuss quickly what would be the number of chromosomes and the DNA content in each stage. Consider a cell which has just entered the interphase. In G1, if we see, chromosome is present as a single thread. So if the chromosome number is 2n, the DNA content would be 2c. This DNA content is doubled in the S phase. So DNA content becomes 4c, but the chromosome number, does it change? No. So the chromosome number remains same, that is 2n. We don't do anything in the G2 stage, so it remains the same, 2n 4c. In prophase, if you see, there is just the condensation of chromosomes, so again we have 2n 4c. In metaphase also, there is just the condensation, and the chromosomes lie along the equator, so we are not doing anything. So the chromosome number is again 2n and the DNA content is 4c. Why? Because two sister chromatids are present. Now let's see anaphase. Anaphase is really tricky. See, there is splitting of the centromere. So there is half of the sister chromatid which is going to one pole, while the other half is going to the opposite pole. So if you see DNA content, so what is happening to the DNA content? It is 2c for one set of chromatids and 2c for the another half. So total DNA content will be 4c. But what's happening to the chromosome number? See, the chromosome number here temporarily will be 4n. Why? Because we have 2n sets of chromosomes which is going to one pole, while 2n is going to the opposite pole. So if we see overall, it will have the chromosome number of 4n and 4c DNA content. This similarly, if we see telophase, we also have the same condition. That's why in certain cells in which there is division of the nucleus, but we don't have a division of the cytoplasm, we get a multinucleate condition, right? We have read about the syncytium, right? So this is all about what happens in karyokinesis. When this karyokinesis, after telophase, when it is followed by cytokinesis, we will get two daughter chromosomes. In one, we will have one set of, the sister chromatids which will have 2c DNA content. Similarly, the 2n chromosome number we will be got. So we will have two copies and each will have a 2n chromosome number and 2c DNA content, because we are just getting half of the chromatid. Is that clear? This is a frequently asked question, like various objective questions will be asked, like what is the chromosome number after the cell has exited the G2 phase, after it has exited the S phase. You can just apply the concept and answer to it. So that's all about today's class. I hope you're clear about the cell cycle, why a cell divides, what are the stages of a cell cycle, what is interphase, what is dividing stage, and you are very clear about the different stages of mitosis, as well as what is the DNA content and the chromosome number in case of the different stages of interphase as well as mitosis. So if you like the video, please like, share and subscribe to my channel. And if you have any questions, please comment down below. I would be very happy to answer them. Thank you everyone. Have a very good day.